All right. Well, hello, everyone, and welcome. I'm Sandy Williams, Gladstone President, and I want to thank all of you for being here tonight. I hope everyone received a program on the way in. I won't repeat some of what's here, but I do want to reference a couple of things that are in the brochure. First, I'd like to point you to the list of names that appear in the first two pages. These are people very important to the operations of Gladstone because they've provided both intellectual input and governance to us, as well as essential financial support. Some of you are here tonight, and I'll thank you in person, but I want all of us to recognize and be grateful to the people who are listed here. Now, the first question I wanted to address with you is, why are you here? You've left a beautiful San Francisco springtime day outside to come into a closed auditorium, but here you are. And you'll, of course, know your own reasons for that, but I'd like to speculate that there are a few things that might have brought you here. First, you may have come to just show support for Gladstone. I suspect that's true for many of you, and I want to thank you for that. It's important that you do stand up for Gladstone. And in that light, I want to comment on some recent events of the past weekend where I and quite a number of other Gladstone people joined with UCSF and other Bay Area institutions for the March on Science, for the Stand Up for Science. So I think you're here, your presence here stands up for Gladstone, but your presence here stands up for science as an important human enterprise. Along with Sam Hawgood and others, I gave a speech to the crowd that day, and I made the following comments about science that I wanted to review again tonight. Science is a noble calling. It's an expression of the human spirit. It's a means to enhance lives, and it's the key to protecting the future of our species and our planet. That's what I deeply believe about science, and I think most, if not all of you, share that with me, and your presence here tonight speaks to that. But I hope you've come for reasons beyond that. I suspect that most, if not all of you, are lifetime learners, and you've come with an opportunity to learn. And I hope at the end of the session today, we'll have fulfilled that expectation on your part. And also, as proponents of science and understanding the scientific method, you know that perhaps the best way to learn is to question. And so we're going to share some information and some perspectives with you, but then we invite your questions, both here in the auditorium for our speakers, and to follow in the breakout sessions for which you'll make a choice among three opportunities. So our goal tonight is to share a bit of Gladstone with you. We want to take you right up to the leading edge of some very important areas of science. And we hope in the learning and questioning process tonight, or this afternoon, you'll go away with a better sense of your own ability to critique developments in the field that we'll be talking about. We want you to feel prepared to tell the difference between the kind of science that really is a legitimate, realistic hope that we can change human life for the better, and distinguish from that from some of the false claims or exaggerated claims that some practitioners might promulgate. Now, to tee up tonight's session, I want to put it in a framework of what's happening in science overall. And I'd submit that there have been three great revolutions that are recent and ongoing that have changed the face of biomedical science. We're adept at using all three of those, but one of those has special meaning here, and that will be the subject of our afternoon. But let me continue with that background and tell you about the three. The first big advance that is accelerating our ability to do science in a way that leads to cures is the revolution in DNA sequencing technologies that make it cheap and relatively inexpensive to acquire vast amounts of genomic information and to understand that information by advanced computational methods. So the ability to and to do this in human beings, in human genomes, and understand how things work using human genetics has been the first of these revolutions. The second of the revolutions is more recent, and I'll mention it especially because it had its origins across the bay at Cal, and this is gene editing. This has been in the news recently a lot. This is CRISPR-Cas9 based technologies and a lot of follow-on technologies that follow that. That's the second of the great revolutions that are giving us unprecedented power to explore the mechanisms of disease and find solutions. And we use that quite adeptly. We're in close relationship with Jennifer Doudna, who's really the founder of that field, and others. But it's the third area of tremendous empowerment that had its origins at Gladstone and for which we are arguably, even unquestionably, among the world's leaders. And that's the field of cellular reprogramming and stem cell biology. One of the great founders of this field is our own Shinya Yamanaka. You've heard him speak at these sessions before. And Shinya and colleagues like Deepak and others have taken the knowledge that we can both not only read the clues as to how a cell decides to be this kind of cell or another, but we can go in and direct that through cellular reprogramming. And that will be, in large part, the general subject area of tonight. So 3 great revolutions, we'll focus on one. Now, our title, Regenerative Medicine and Stem Cells, A Promising Horizon, harkens back to what I said at the first. The promising horizon is such that is our way of stating that emerging science provides a realistic hope that currently unsolved diseases can indeed be conquered and mastered. And we believe this is true because it's already happened, at least in part. Biomedical research hasn't improved the quality of life, extended life for people all around the world. Smallpox and polio have been eradicated. Deaths from heart attacks and strokes have been drastically reduced. HIV is no longer a death sentence. Gladstone scientists had a hand in creating the foundational knowledge that led to many of those advances. But as you know, there's much more to be accomplished. There are cruel and common diseases that remain to be solved, and Gladstone science points to that end. It is that goal that energizes and sustains us every day. Now, tonight we're going to hear from 2 of Gladstone's scientists. I won't give a detailed introduction. Please read about them in the program. But you're going to hear from Deepak Srivastava, who heads our Cardiovascular Institute and has been, as I mentioned, one of the true pioneers in the application, in the study of how the heart develops and how we can use that knowledge to control that for therapeutic purposes. And then I think new to this forum, though, will be Li Gan. Li Gan is a neuroscientist who was recently honored by promotion to be the Associate Director of our Neurological Institute. And Li will talk to you about the way some of this emerging science and technology is altering our approaches to neurological diseases. So, Deepak?
I'm going to use one of this so that I can get it away from behind the podium. So I'm delighted to see so many familiar faces in the audience. Thank you for being here. I think based on the revolutions that Sandy mentioned, and because of advances in the technology industry, mathematics, computation, I think it's safe to say, and it's not hyperbole, that we sit at a moment in time in mankind's history where we have the opportunity to understand human disease and biology at a depth that we've never been able to before. And it's our premise that it's that understanding that's going to lead to more rational design of therapies in the future compared to what we've been doing in the past, which has worked on occasions. But we've been, in science and medicine, we've been trying to develop therapies for a variety of diseases, but without full knowledge of what is actually going wrong in the setting of human disease. We didn't have access to human cells before. We couldn't manipulate the genome of human cells before, and we didn't even know what mutations were causing disease until recently. But we can do all those things now. And that's why this is really a moment in time where I think over the next 10 to 20 years, we will see advances in medicine that far outpace the advances we've had over the last 10 to 20 years, which have been significant, but relatively incremental. And I think at the core a lot of this ability to understand human disease is to finally be able to understand a human cell in a way that we couldn't before. And what we've been able to do in understanding a cell and how it goes right and how it goes wrong has largely been informed through the study of stem cells, and that will be the subject of today's discussions. And so why do I say that? The reason is that a stem cell can turn into any different cell type in the body. And so if you understand how that happens, then you understand how a cell is being controlled. And the discovery that Sandy mentioned that Shinya Yamanaka made, which was essentially that we could take an adult cell that already knew what it was and reprogram that and change it so fundamentally that it would now behave just like an embryonic stem cell that could again become all the different cell types of your body, fundamentally changed how we thought about cells in our body and how we could manipulate those, because it taught us that, in fact, if you knew enough about that cell, you could control it almost at will and make it do what you want it to do. And if you understand that, then you might understand how a disease could hijack a cell and make it a disease cell. And if you knew that, boy, then you know how to take it back, right? So it's the fundamental aspect of all of this is understanding how a cell decides what it's going to be and how it will respond to various stresses that might be imposed either by a genetic mutation that causes disease or by environmental influences that we're always getting that then result in disease. So with that backdrop, before we talk about stem cells, let me make sure we're all on the same page about what that means. So a true stem cell in your body has the property that it can become any different cell type that's present in you. That's one property, and there's only a second property, there's no more. The second property is that it can divide indefinitely, so it can make more and more of itself forever. That's it. That's an embryonic stem cell, which is a true stem cell, has those two properties. There are adult stem cells in all of our bodies that are also very important, that help replenish cells within one organ. And those are stem cells, but they can't turn into all cell types in the body, but they can keep rejuvenating an organ. And there are many organs in your body that do have that rejuvenation over time. And the blood is the most famous of those. And blood stem cells are the ones that are used for bone marrow transplants. So we've been doing that for a long time, and it's very successful. For most other organs in your body, though, you can't really take those out, grow them, and put them back in. And so we've had limited success with adult stem cells in other parts of the body. Now, Sandy mentioned that there are types of treatments that some are doing, even in this country, but a lot more around the world, that haven't been validated. And I thought I'd mention that because there are a lot of stem cell treatments that you'll read about, and many of you in the audience have asked me about for yourselves, that you look online and people are saying that this can cure virtually every ailment under the sun. Unfortunately, most of those have not been validated. There's probably not a lot of great science behind that, and it's premature. And so what we're trying to do here is to take this step by step in a logical, scientific fashion and understand these cells carefully and then utilize that knowledge to address disease. And it's still early, but it's happening. So if I've told you that a stem cell, an embryonic stem cell in particular, can become all the different cell types in your body, yet the DNA code is exactly the same in every cell, how does that happen? It turns out that we have a fairly deep understanding of that now, and essentially the DNA is the same in every cell, so you can think of that as the hardware. But the way that DNA is read, imagine that as a software, it being that what genes a cell decides to turn on and what genes it decides to turn off and have inactive is really the key. So you may have 20,000 genes in each cell. So the DNA encodes for genes that encode for protein. If each cell has about 20,000, in any given cell in your body, say a heart cell or brain cell, only a subset is active, maybe 5,000. That means 20,000 genes are silenced. That essentially is the key. If you can control which 5,000 are on at any given time and which 20,000 are off, you can change the fate of a cell by just flipping all those switches and alter what it is. The same thing happens in disease. Different genes get turned on and off that should not be turned on and off. If you can figure that out and flip those switches back to normal, you can stop disease in its tracks. And so we are beginning to understand that all through the study of stem cells, and now we finally have the opportunity to, I think, get at the core of human disease, and that's what we're doing, particularly here at Gladstone. So, an iPS cell, you've all have heard about these induced pluripotent stem cells that Shinya's lab discovered years ago. That technology can be used in multiple different ways. So let me just summarize those briefly. The first is to use those cells to finally have in front of us human cells that have disease or have a disease mutation. And we at Gladstone are doing that for a variety of heart diseases, brain diseases, and others. And now we can generate billions of human cells, interrogate those in a way we never could before, and then find out what's gone wrong, and then screen for drugs that could correct that. That is happening right now here and across the world. And in fact, we're at the point now the field has evolved where we've not only understood the mechanism of disease using human cells, we have screened for drugs, we have candidate drugs that now we're testing in various animal models, and that's going to lead to those clinical trials with those. So we're really getting close to now many of these being in clinical trials that have been discovered with iPS cells. That's one approach that's happening. The second is taking these cells, turning them into what cell has been lost in your body that's causing your disease, and then now trying to transplant those cells back into the patient to treat that disease. So that too has advanced, and last year, the first patients were treated with an iPS-derived cell that was transplanted back into the patient. It was identical to that patient because you can take a cell from the adult, transform it, transplant it back. There's no issue of rejection. That has happened for macular degeneration, an eye disease that causes blindness, and that patient is a year out now and is doing well and is being monitored for improvements. It was a safety trial, obviously, but there's some signs that there may even be some improvement, and those trials are now going to continue. Now, there are many other areas that'll happen in the coming years, including Parkinson's, diabetes, and other areas, and those are all, and spinal cord injury, and those are all following. Now, the third area is a different one, and that is one where, as I mentioned, we can modify cells, we can engineer cells because we understand them well enough. So here, the area that's really at the forefront is related to your T cells. So T cells are part of your immune system. What we can do now is make your iPS cells, turn them into T cells that are part of your immune system, manipulate those, modify those so that those cells now can recognize bad things in your body. We put them back in you and they can go and attack those. The most famous of those is in the cancer field, where people have been able to manipulate those T cells so that they can recognize cancer cells in your body that have otherwise been under the radar. Cancer cells are very smart. They create this almost shield around them so that your immune system can't see them. That's why they can grow in you. Otherwise, your immune system would kick them out. So what we've done here is we've made your T cells, engineered them so the cancer cells are no longer invisible. they're recognized by your T cells. We put them back in you, and they go and find the cancer cells in your body and kill them. It's a revolution that's going on in the cancer field right now. We think that that same approach can be utilized not just to fight cancer, but to do a number of other things to attack disease where your immune system could go in and do the right things in controlling inflammation or affecting a tissue by altering autoimmune diseases and things like that. And you'll hear more from Li Gan in a moment, both about the use of transplant approaches and immune approaches to try to tackle neurological disorders. So I want to turn now for a moment to this notion that you can control cells to be what you want them to be. I've told you that we can take your cells and turn them into a stem cell that then we can then redirect into, say, a heart, brain, pancreas cell, et cetera. But what if we knew enough so that we didn't even have to take this circuitous route and take your cell and make it a stem cell and then, say, a heart cell, but rather we could go directly from your cell that you already have as an adult and directly turn it into a heart cell or a brain cell or a liver cell without it ever even becoming a stem cell. So now we can do that. We have found ways here at Gladstone, I think we've been the pioneers in this area, which we often refer to as direct reprogramming because it goes directly from one cell type to another. We can turn cells in your body directly into heart cells, brain cells, liver cells, pancreatic cells, just by knowing which switches to flip and what cues to give, without them ever becoming a stem cell. So the reason that's powerful is that for some organs, there are a number of cells in the organ that you could harness for regenerating that damaged organ. So let me explain this. The heart, for example, you think of it as a beating muscle, right? They're beating cells in the heart that make it pump. It turns out that only half the cells in your heart are muscle. The other half in your heart are just support cells for that muscle. They form the architecture of a scaffold. And they're there, they're important, but they're not muscle. They're never going to be muscle. So when somebody has a heart attack, your muscle cells die. Those support cells actually go to form the scar so your heart doesn't explode open, and you kind of heal that with scar tissue. Well, what if you could convert those cells that are already in the organ? They're there already. Give them the right cues, so now they became beating heart muscle. You could regenerate the organ from within with your own cells. So that's what we've been able to do over the last few years by sending just a few key cues into the heart after damage. We can convert those support cells into new beating muscle that integrates with the other muscle cells in the heart and beat in synchrony and actually can increase the amount of blood pumped from the heart. We've done this in mouse, and then we've advanced this. We can do it in human cells in a dish, and most recently we've been able to do it in large animals, particularly pigs, whose hearts are more similar to ours in size. And there we can improve the heart function. So that technology has advanced pretty far. It's not quite ready for a clinical trial yet, but the path is clear. And so we've recently licensed that out into a startup company, a spin-out company from Gladstone, which we've called Tenaya Therapeutics. And that entity, biotech company, will now take that and develop it so that it can be tested in a clinical trial. We think that approach is not unique to the heart. We should be able to do that for the number of other organs that I mentioned, because it's really just knowing the cues that are telling that cell what to be. So we think it'll be a broadly applicable approach for a variety of diseases where you've lost cells, you just need to get new ones. Now, I'll close with what we've done most recently here at Gladstone, which is another alternative approach to switching a cell's fate, but also gets to the stem of being able to control what a cell is doing. So many organs in your body, once you become a few weeks after birth, the cells that you have in that organ is what you got. And that's why if you have a heart attack or if you have a stroke, then you lose cells, you don't really get that many cells back because those organs don't have great adult stem cells in them to replenish itself. But typically, you don't lose all your cells. There's some cells left, whether you have a heart attack or a stroke. So what if you could get the cells that are left and are good to just make more of themselves? Meaning, can you get them to divide and replicate and create more of themselves? That would unleash the potential for you to regenerate again from within. And so we've worked really hard the last few years to find a way to unlock the ability of a cell to divide, because after all, when we were all embryos, all our cells were dividing all the time. That's how we grew. That's how we went from, that big to this big. So all we have to do is figure out how nature does that in an embryo and redeploy that again in the adult, and maybe we could make that happen. So just in the last year, we've actually been able to do that. We've found the right cocktail of factors that can tell an adult cell to go back in time and be more like an embryo and divide again. We've applied that to the heart, where we've been able to regenerate heart muscle. So far, just in mice. Now we're going to advance that into to larger animals. But we can create a lot of more new muscle by doing this. We've tested it so far in brain cells. We think we're close. It does it pretty well there, but we've got a few more steps that we think we're missing. We've tested it in the ear. Why the ear? So when people lose their hearing, there are neurons in the ear that are actually sensing vibrations. And typically in hearing loss, those are the ones that are lost and you don't get any more. I have a personal reason for testing this. Even though I'm a cardiologist, I have twins who are 15 years old, and they both have hearing loss in both ears. They wear hearing aids. They do fine. But it sure would be nice if we could get them some more of these neurons so they didn't have to wear those. So we've tested that, and we think we're very close there also to being able to get those cells to divide. And we're testing a variety of cells in the body, but again, we think this will be a broadly applicable approach to multiple different cells in your body to try to regenerate more. And so what all this, I think, really starts from is having a very fundamental and basic understanding of how a cell works. Because if you can figure that out, then you can manipulate the system in order to fix the problems. And if you don't understand that, you're just shooting darts. And so at Gladstone, our approach is basic science, and it's really getting at the core of how cells normally and in disease works. And it's our belief that, and I think we're demonstrating it, that is the key to being able to address human disease. And I've given you a couple of examples of those, and there are many, many others, great science going on in the institution, but it all starts from there, a basic understanding of the cell. So with that, I'm going to stop and introduce Li Gan, who's going to give you some examples of some of these approaches, specifically in the brain around Alzheimer's disease. Li?
So thank you. Thank you for coming in such on such a beautiful day. And I want to follow up with what Deepak has told you about the enormous potential of regenerative medicine that has changed many disease fields. And what I will focus on is brain disease called, in particular, Alzheimer's disease. So you heard from the news, Alzheimer's disease has gained a lot of media attention. It is the most common form of dementia in the elderly. And even just in the United States alone, there are 5 million of the Alzheimer's patients. And this is a devastating disease. It robs people of their memory and as well as the ability to think. And the reason for that is because the regions that are responsible for those functions, such as memory, new memory formation, are severely damaged in the AD patients. So there are currently no treatment that can slow down the progression of the disease. And you may have heard from the news, there are more recent failed clinical trials And partially, or there are many explanations, but partially is that this treatment may not be able to reverse or repair the existing damages that have already occurred in these patients' brains. So can we harness the power of stem cell therapy to treat AD? So before I go into that, so I will tell you two potential approaches that have been being actively pursued at Gladstone. But before I go into that, I want to remind you that how complicated our brains are. So it is, arguably the most complicated and least understood organ in our body. Maybe heart is another one.
Heart is simple.
And just there are 100 billion neurons in our brain, and 10 to 50 fold more of the glia cells, supporting cells, more than the neurons. And these 100 billion neurons form massive communication networks that are constantly shaped by experience, such as formation of new memory. So as you hear me speak, the information is processed by the neural networks, and also the new memory is formed. And in the AD patients, these networks or these connections are lost, and many neurons are lost as well. So the traditional wisdom is that it would be very difficult to use stem cell therapy, in this case, cell replacement therapy, to treat AD. Because you can imagine how you can place these large numbers of neurons at the right place and form the right connections. So the two approaches I'm going to mention to you is challenge this traditional wisdom. And I think we believe the key is to select the right type of cells that may overcome some of the limitations. And the first approach is a type of cell called interneurons. So interneurons, there are many, many subtypes of neurons in our brain, and interneurons is one type of it. It accounts for a very small percentage of total neuron population, but it has tremendous effects on the neural network. And the reason for that is one single interneuron directly communicates to 3,000 or more of other neurons. And they modulate their activity. So to use an analogy, interneurons are like conductors in a symphony. So a very small number of them can actually have a profound effect on the network function as well as brain function. So actually, several labs at Gladstone have made important discoveries to find that interneurons are dysfunctional in AD brains, and the dysfunction and loss of the interneurons in the AD brains play an important role to the cognitive decline in the AD related dementia. So more recently, Yadong Huang's lab at Gladstone has reprogrammed using the power of the iPSC technology to reprogram stem cells, human stem cells, into interneurons. They have actually introduced these reprogrammed interneurons into AD mouse models, and they put in a region that's responsible for the memory called hippocampus. And they found that in their initial analysis is quite encouraging. So they found that not only those reprogrammed interneurons can replenish the lost interneurons in these very old AD mouse models, they can also improve the learning and memory of these mice. So they are continue to test this in animal models, expanding the repertoire of readouts. But the ultimate goal is to hope to introduce these reprogrammed human interneurons into AD patients once we know more about it in clinical trials. The second approach I want to discuss is to follow up on what Deepak had told you in the cancer field, is to restore the immune system in the brain using stem cell technology. So we know that immune system in our body are our body's defense mechanism. And brain's immune cells are called microglia. They defend, so like other immune cells, they defend our brain from invaders such as infections, but also the buildup of protein aggregates that often occur in the aging brain. They account for a very small percentage of brain cells, but they are enormously important in maintaining a healthy brain environment. So can we build, so in AD brains, actually, the immune system in our brain is not working well. Microglia are dysfunctional, so that's why we have the buildup of protein aggregates, in particular, like amyloid plaques. They also have turned on the toxic expression of toxic factors that actually causing collateral damage to the surrounding neurons. So can we restore or rebuild brain's defense mechanism in AD? There are reasons to be optimistic. And the reason is that microglia are actually programmed to go to where injury sites are. In other words, they seem to have an internal GPS system to tell them where they go, where they're mostly needed, because that's how they are programmed. Another reason to be optimistic is microglia as a type of cell that's known to secrete factors or release factors that influence the neighboring cell. That's how they become toxic when toxic factors are turned on. So for these reasons, that we think that it is possible to have reprogrammed microglia to rebuild the defense system. So how do we go about it? We have established a very efficient platform that allows us to reprogram human stem cells into microglia. So we have put those reprogrammed microglia into the brain of AD mouse models. So we are testing whether those new immune cells will be beneficial. But in the meantime, we are taking advantage of this new genomic editing technology to build designer microglia from the human stem cells by taking out the toxic factors that get turned on during the AD, but enhancing the genes that are truly functional, important for the function of microglia. So we're hoping that this designer microglia can actually help restore the defense system in the brain. So these are early days, and we have long road ahead of us. But I'm very excited to share some thoughts with you today about the enormous potential of using the technology to treat AD and other brain diseases. And I'm happy to answer questions during Q&A and also in the breakout sessions.
Deepak and Lee sit here and have the handheld microphones, and I'll moderate from up here. We're going to open this up for questions from you. Before we do that, though, I thought in the interest of the learning opportunity tonight, I might try to summarize what I think I learned from what we just heard. If you were listening carefully, you heard about human genetics informing our ability to control what cells do. You heard about gene editing, determining our ability of how to take cells and make them do special things. But you also heard that much of this was grounded in our ability to understand how to tell a cell what kind of cell to be and how to control how many numbers of those cells are present at a given time. We are gaining command of those important properties through the basic science that's being done here. And then that then leads to three approaches that would cure diseases. One is to create authentic models of human diseases, heart diseases, brain diseases, others, in a dish so we can have greater predictive value whether a drug candidate will actually work in a human than we've ever had before. The second is to create cells outside of the body that we would then put into the body for a therapeutic purpose, regeneration or otherwise. And the third is not to put cells into the body, but to send signals to cells that are already there to make them pursue a course that they otherwise wouldn't do on their own that would promote health. So that's, I think, the common denominator of what you've heard. And then there was one more really interesting part I hope you picked up on. Some of you may not have realized that the brain has its own intrinsic immune system, and that those cells are very special. They resemble immune cells elsewhere in their body, but there's a special kind. And in the same way that physicians are harnessing the power of circulating cells in the body to eradicate cancers, it may be possible to harness the power of the brain's immune system to clean up things that otherwise destroy the functions of brains. So that's kind of what I learned from this, and I hope you learned more. But now let me open this up to questions. We'll have questions of Li and Deepak here for a while, and then I'll describe what the breakout sessions will do that we'll move to in a bit. Yes, in the back.
Hi, I think you can hear me.
Yeah.
Oh, I'm sorry. I apologize. Hi.
Hello, Chuck.
Standing here as I see a lot of other gray-haired people, and being a businessman who's always interested in how long it goes to take something to be completed, I'm very concerned about the fact that other governments, such as China, France, Germany, Switzerland, are more exponential in their moving ahead with approving some of these things than the United States. And so I'm just wondering your thoughts on that and how we move ahead more exponentially in the United States, and what is the time frame on a given drug solution once it gets beyond the mouse stage at the various stages. How long does it normally take for something from the mouse forward to get approved for human trial? Thank you.
Well, I think that's it's a really important point, and I think what you're getting at in the first part is regulatory hurdles that might be present, and could we do something more creative? And you are correct that there are a number of countries, particularly Japan, that has taken more creative approaches in recent times to allow drugs to advance into people, particularly for diseases where there's no other therapy, and then gather that information over time while a clinical trial is going on. And I think our government is considering those options also. I know our FDA is. We have to balance that with two things. One is safety, of course, which is being careful that we're not doing things that could be damaging to people without knowing. But the other complication in our system is that if you approve a drug that for treatment when you don't know if it works yet or not, particularly for those who don't have any other options, that makes sense. But then who pays for it? So should an insurance company then pay the high cost that it might be for that drug when it may or may not work, but a patient might expect that, and then if they don't, then you're saying that, well, if you have enough money, then go for it, but otherwise too bad. So it's not a simple problem, but it's an important one to tackle, and I think people are grappling with that right now.
Dr. Srivastava, what are the prospects, do you think, for replacing the device known as a pacemaker inserted to ensure regular heartbeats in a patient whose innate pacemaker in the right atrium has begun to fail, so that there are pauses between heartbeats that become concerning enough to lead to the implantation of a pacemaker? And will that take the form of some biological restoration or rejuvenation of the failing pacemaker cells? Or will you think it will be transforming perhaps adjacent cells that are not currently pacemaker cells into becoming replacement pacemaker cells, or perhaps exogenous cells, stem cells, what have you, that have been differentiated into those cells with those magical properties of a pacemaker? And if it's the latter, how are you going to avoid the development of multiple pacemakers with all of the consequences that might happen?
Deepak, that's in your alley.
Sure. The issue here is that there are people whose hearts, the pacemaker in the heart, which tells the heart to fire at a certain rate, starts to fail. And we do fortunately have a pretty good medical treatment for that, which is to implant a mechanical pacemaker. A wire goes into your heart, and the wire sends the stimulus, and that becomes your pacemaker. But there are obvious problems with that, and sometimes they stop working. So that is a problem we've actually been tackling in our lab for the last several years. We have begun to understand what it is that tells a pacemaker cell to become a pacemaker cell for the reasons you mentioned. And so we've begun to crack that, and we think that you're right, it may be possible to convert cells in one tiny area in the heart into new pacemaker cells or even transplant them. The beauty there is that these are very discrete, small number of cells that form the pacemaker. So the challenge of regenerating or replacing those is much more tractable than creating tons of new muscle. So in fact, the approach you mentioned may be a leader in some of our therapies because it's a small number of cells we have to tackle. So we are working on that. I didn't answer the second part of your question, Chuck, which is how long does it take? It still takes a long time, but that time frame is being compressed rapidly because of technologies. But it's still, from a mouse to testing things in large animals takes a few years. From there, it's a few more years in a clinical trial, and then that process takes a number of years. So I would still say from the time if a discovery is made in mouse, if you're aggressive, then you're still looking at about 10 years.
There's an important point here that I think connects the cardiovascular field with the neurological field. In the problem of heart failure, which is one of the targets that we attack, it's a big unsolved problem in cardiology. Correction of that problem is going to require making 1 billion or more new cells. So it's a real numbers problem, unlike the problem of a pacemaker where you may only need a few hundred. So that's the case. What Lee was mentioning is when we thought about the brain with its 100 billion neurons, to fix a damaged brain by putting new cells there, if it requires billions, that's a very formidable procedure. But what Lee has pointed out, and I'll let her elaborate, is these interneurons are such special cells that it may be possible to help brain function quite a bit with a very small number of cells. Lee?
Yeah, I mean, I think one of the challenges is to have enough cells to be at the right place and form the right connection. And some of the cells that Gladstone scientists are focusing on are the ones that a small number would have a big impact. And these include the interneurons, because one can innovate or communicate with 3,000 or more. And another is the immune cells also, although they're small numbers, but they have actually huge impact. So those are the type of cells could hold promise and could be further developed into feasible cell replacement therapy. But understanding how they work is important.
Bruce?
I think Jojo.
Oh, okay.
First of all, thank you very much. My husband is close to 99, and he has been diagnosed with Alzheimer's and dementia since 2003. Deepak knows him. He is in the Pacific Union bridge tournament and winning with men who are 50 to 70 years old. He speaks perfect French. He speaks wonderful Italian, and he doesn't understand anymore. He does recognize me. I spoke to his neurologist. The cost of medicines at this point are about $700 a month. And I've been monitoring that for the last 6 to 9 months, and I don't see any help that these medicines are giving him. I asked his neurologist, and the neurologist said to me, well, we have to put that on the chart, and they have to give it to him. Otherwise, you could sue me for malpractice. And this is very alarming, and I would love to hear a little bit more about that because, Doctor, you said there's no way to slow it, and I have lived with no way to slow it. And thank you very much.
So I think right now what we heard from the neurologist is that the current drugs really do not have efficacy. Sometimes in some small number of patients it can help symptomatically, but the reason for these drugs to be not able to help is because they do not tackle the mechanism, like why the progressiveness of the dementia. So I think the past decades, there are a lot of new mechanisms that have been discovered, and there are drugs that's been developed, and some of them with the hope to slow down what causes these neurons to become dysfunctional. What I have discussed today is a next generation of potential therapy that can actually go there and repair or restore the lost neurons and connections. So I think because Alzheimer's disease is such a complicated, multifactorial disease, it is likely that in the future there will be combination therapy that's needed to slow down or stop or even reverse the progression. So I think, like Deepak said, understanding the mechanism, how they progress, is the key, and that's what we are working on.
I might add, the Alzheimer's Association estimates that by mid-century 2050, in the U.S. alone, we'll need to spend $1.5 trillion a year just for the care of patients with Alzheimer's and other dementias. That's in the absence of any disease-modifying therapy. If we have effective disease-modifying therapies, we'll halt the tragic human cost of this disease, but also save our health systems a lot of money that can be redeployed elsewhere.
Let me just say that the story you told really gets to the heart of the issue that we're talking about today, which is that for far too many human diseases, we don't have any good medicines. And virtually every Alzheimer's trial to date has failed. For heart disease, we haven't had a new medicine that addresses heart failure in 25 years that really makes a big difference. And this is for heart disease. I didn't mention there, if you have end-stage heart failure, your 2-year mortality is 50%. It's as bad as most cancers. We've got nothing other than a heart transplant. And so the reason we think is that all these trials have failed because we didn't understand the system properly. But now, today, we have the ability to do that, and that's why we're focused there.
Enjoyed the presentations, thanks. Deepak, I had a question for you. The liver is an example of an organ which can regenerate, but it doesn't always get it right. So with acute liver failure, it comes back quite nicely if you can keep the patient alive. Chronic liver disease, not so much. We get disordered regeneration, portal hypertension. Could you just comment on how that plays into your thinking about how you would regenerate an organ and preserve sort of the architectural framework in a way it still functions?
Yeah, I think that's important. The architecture is really important to the function of most organs. It's one reason that we have thought that converting cells in your body while they're there already might be a better approach than introducing exogenous cells, which then would have to somehow form the right architecture. At least in our experience so far in now multiple organs from our group and others, when cells convert in their native environment, they're getting the cues that we don't yet fully understand from their neighbors that are telling them where to be and how to be in the right place. There's a whole new field of what we call organoids, where in a dish with stem cells, we can let cells aggregate and they start to form proper three-dimensional structures, mimicking organs. It's really remarkable. What that tells us is that there's intrinsic knowledge in our cells of how they should interact with one another and shape themselves that we're far from understanding. But these organoids are, I think, if we dissect, should be able to understand how the cells know what to do properly. But I think exactly what you described in the liver is a case where sometimes that fails. And we have to learn it better.
There's a point I'd like to make here. There's a lot of very wishful thinking in the stem cell field. Deepak alluded to the fact that, particularly in other countries, but even in our country, clinics have sprung up to inject people with various kinds of so-called stem cells. For the most part, much of this is premature. The cells that are being utilized are poorly characterized, they're poorly understood. It's a kind of let's try it and see what happens approach, despite the fact that there'll be testimonials from some patients who believe they've been helped by this, but it's really unproven and poorly founded. What you're hearing from our scientists is a much more systematic and careful approach where we're going to demand a degree of understanding of what we're doing and exert tremendous quality control over whatever procedures we introduce into people, first to be sure they're safe, and then to be sure that we have rigorous tests of how well they're working.
My question's for Sandy, actually, and it relates to the first question of the symposium. And there was an interesting article by Raphael Reif, who is the president of MIT, recently addressing the whole issue about the reduced funding for basic research, not applied research, but basic research. And also he's been very outspoken about the whole immigration policies of the current administration. My question really relates to sort of the position Gladstone is taking with regard to advocacy for scientific funding and also for the need for a broader engagement or embracement of brilliant scientists, immigrant scientists into our research communities.
Yeah, thank you for that question. I met with the MIT president back in January, and we had conversation on this. Our scientific enterprise is under threat from the statements made by our president about reducing funding to the NIH and other science organizations. This would be a devastating action to the science community, particularly the basic science community, since much of the original discovery that's the raw material for cures depends upon NIH funding. So there's a storm cloud on that horizon. However, there are people on both sides of the aisle in Washington, including some prominent Republicans whom we know and are in communication with, who've stood up for the NIH. And the bill passed this week for the 2017 budget is actually quite favorable. So I don't think a cure for Alzheimer's disease ought to be a partisan issue. And I think we need friends from everywhere to stand up for science, for the value of evidence in policymaking, and for a strong NIH as the core of what will drive the future cures. That alone won't do it, and organizations like Gladstone use a variety of funding streams. We're about 50 to 60% NIH supported. We're about 15% supported by various partnerships with industry groups in the later stages. And the rest of our funding comes from our trust and from philanthropy. So it's a delicately balanced ecosystem. We're doing fine now. We can survive and prosper at Gladstone if there are modest cuts to the NIH, though that'll be bad for the country. If they're really deep cuts, it'll be an existential issue for places like Gladstone and others like us. So it's an important public policy issue.
And I think it's worth recognizing that based on what you've heard today, I think you can tell that we are at an inflection point in our ability to tackle these things. And it's just at the wrong time that the commitment to advancing biomedical research is waning. And even before this administration, over the last decade, in real dollars, the amount our country spends on biomedical research is down 25% in real dollars. Just at this time of opportunity is on a significant upstroke, the investment is declining. And so I think that's something we need to think a lot about as a nation, given that we spend 16, 17% of our GDP on healthcare. We should decide what the incremental percent we should spend as an investment to address those costs, because there's an argument to be made that these investments will pay off in terms of lower costs for our society as a whole over time.
Yeah, we spend the highest proportion of GDP on healthcare of any other nation, but we spend a lower proportion on medical R&D. And U.S. has been a magnet for talent from all over the world to come join our biomedical enterprise. That flow of talent is in danger of being reversed if we can't maintain strong public as well as private support for biomedical research. You asked about advocacy. We are advocating through our organizations, the National Academies, Research America, the American Association for Advancement of Science, as well as my wife Jennifer's here. We were marching on the street last Saturday, so it's time that scientists come out of their labs and speak up.
Go to breakout sessions?
Yes. So the format tonight wanted to break us up into three thematic sessions. So we'll switch to that mode now. Those of you who want to ask about brains, stay in this room. And Lee will be joined by other brain experts to answer your questions. Please move down to the front so it's more intimate. Those who want to ask about hearts can join Deepak and some of the other stalwarts from our cardiovascular program in the conference room that's immediately adjacent to the auditorium here. And then for those of you who may be somewhat lacking in judgment, the third option is to ask the president. And I will be in the conference room around on the other side facing the lobby. My hope would be is you might filter yourselves out before you get to me, but I will be there if anybody would choose that option. So thank you, and then after all that's done, we'll gather for some refreshment in the lobby. So thank you.